Hello, dear students. Today we are going to speak about setting up a nerve impulse. Let's start. Investigating nerve impulses. Nerves convey information rapidly from one part of the body to another, enabling animals to respond quickly to changes in their external and internal environments. The information is carried in the form of electrical signals called nerve impulses. Most of our understanding of the nature of nerve impulses comes from work done on giant axons of squids. These are the nerve fibers responsible for the rapid escape movements of squids. Their large diameter makes it possible to measure the electrical activity in a giant axon when it is at rest and when it is conveying a nerve impulse. A fine glass microelectrode is inserted inside an axon, and the voltage between it and the reference electrode on the surface of the axon can be displayed on a cathode ray oscilloscope. By convention, the potential difference of the inside of the cell is always measured relative to that on the outside, so that the outside potential is taken as zero. Resting potential. A resting neuron is so called because it does not convey a nerve impulse, not because it is inactive. On the contrary, a resting neuron expends much energy in maintaining a potential difference across its membrane. This is called the resting potential and measures about 70 millivolts. During the resting potential, the inside of the neuron is negative relative to the outside because of an unequal distribution of charged ions. On the outside, sodium ions, chloride ions, and calcium ions are present in higher concentrations than inside the cell. By contrast, the inside of the cell has a higher concentration of potassium ions and organic anions. This unequal distribution of ions results from a combination of active transport and diffusion of sodium and potassium ions across the cell membrane and the inability of large organic anions to pass out of the cell. A sodium-potassium pump actively transports sodium ions out of the neuron and potassium ions in. For every 3 sodium ions pumped out, only 2 potassium ions are pumped inwards. On its own, this would result in only a slight potential difference across the membrane. However, this difference is amplified by the membrane being about 50 times more permeable to potassium ions than to sodium ions. Potassium ions are able to diffuse freely back out of the cell down their concentration gradient, but the sodium ions diffuse back into the cell only very slowly. This creates a negative electrical charge inside compared with outside. Without an active transport, an equilibrium would eventually be reached and there would be no potential difference across the membrane. Action potential. A nerve impulse occurs when the resting potential across the membrane of a neuron has a sufficiently high stimulus. A stimulus is any disturbance in the external or internal environment which changes the potential difference across a membrane. The stimulus may be chemical, mechanical, thermal, or electrical, or it may be a change in light intensity. The recording on the cathode ray oscilloscope shows the effects of a stimulus on a giant axon. When the stimulus is applied, the axon becomes depolarized, that is, the inside becomes temporarily less negative. If the stimulus is strong enough, an action potential occurs. There is a complete reversal of the charge across the nerve cell. The interior becomes positively charged relative to the outside. Typically, the action potential reaches a peak of about plus 35 millivolts. The potential difference then drops back down, undershoots the resting potential and finally returns to it. The return of the potential difference towards the resting potential is called repolarization. The entire action potential takes about 7 milliseconds. Although this example refers specifically to a giant axon, its general features apply to all animal neurons. Ion channels and action potentials. The action potential results from the changes in the permeability of cell membranes to ions. At rest, the membrane permeability of a nerve fiber is thought to depend on ion channels through which specific ions can move. An ion channel consists of a protein molecule spanning the membrane with a pore through the center. Sodium ions move through one type of channel and potassium ions through another. There are many more of these ion channels for potassium than for sodium. Therefore, at rest, the membrane permeability to potassium ions is much greater than that to sodium ions. During an action potential, special ion channels control ion movements across the membrane. These channels are believed to have voltage-sensitive gates that open and close in response to voltage changes, and are therefore called voltage-gated ion channels. During the resting potential, the voltage-gated sodium and potassium ion channels are closed. When a stimulus is applied, sodium ion channels open rapidly, sodium ions move in, and the inside becomes more positive. If the stimulus reaches the threshold level, an action potential occurs. When the action potential reaches its peak, the sodium ion channels close slowly and potassium ion channels open slowly. Sodium ions stop moving into the cell, but potassium ions diffuse more rapidly out. These changes cause the potential difference to drop. When the membrane returns to its resting potential, potassium ion channels close, but because they do this slowly, the potential dips below the resting level. Finally, when the potassium ion channels are closed, the membrane returns to its resting condition. That is the end of our video session. In the next video, we're going to speak about homeostasis. Thank you for watching.